A lab makes a drug that blocks one thing. 1. It sits on a single receptor and stops one molecule from binding to it.
Go on.
They put it on smooth muscle cells, stimulate them, and measure 5 enzymes. 4 of the 5 don't move. 1 drops.
Okay. So, which one?
That's the question. And here's what makes it good. The enzyme that drops is not the one the drug touched. The drug never went near it.
So, the drug hits one place and the damage shows up somewhere else.
Somewhere else entirely. And if you know the architecture, you can point at it before you finish reading the question.
Hello, I'm Yue. This is the USMLE reasoning lab, where we don't memorize answers. We build the logic until the answer has nowhere left to hide.
And I'm Maya. Today is a long one, and it's on purpose.
If you've ever memorized this pathway 3 times and lost it 3 times, subscribe. This is the episode that makes it stop happening.
Let me guess what's in your head right now. Alpha, 1, then GQ, then phospholipase C, then IP3 and DAG, then calcium.
Honestly, that's exactly what's in my head.
And you'll lose it by Thursday.
Probably by Wednesday.
You'll lose it because that's a list of names joined by arrows. There's nothing underneath it. You can't rebuild an arrow you've forgotten. So once one link goes, the whole chain goes.
What's the alternative then?
We stop drawing arrows and start naming objects. What is each of these things? Physically, how many separate molecules are we actually talking about? Where does each one sit? Which piece does the work?
And once I know that, the arrows come back on their own?
They stop being arrows. They become the only thing that could possibly happen next. That's the difference between remembering a pathway and being unable to forget it.
All right. Where do we start?
With a parts list. Nothing moves until every part has a name and an address. First correction. And it's the one that fixes the most damage. When people say G protein coupled receptor, they picture one thing, one blob in the membrane that does everything.
That is what I picture.
There are 3 separate players sitting in 3 different places, and most of the time they aren't even touching each other. I'll say players rather than proteins because one of the 3 turns out to be a team.
3. Give me all 3.
Part 1, the receptor. It sits inside the membrane and goes all the way through. Part 2, the G protein, which is stuck to the inner face of the membrane but doesn't go through it. Part 3, the effector, which is an enzyme, and it's the one that actually manufactures something.
So, the receptor isn't the thing that makes the product.
The receptor never makes anything. Not once, not ever. Hold on to that, because half the wrong answers in this topic come from forgetting it.
Let's take them one at a time.
One at a time. Part 1. The receptor is a single chain of amino acids. 1 protein, 1 chain.
And it crosses the membrane 7 times.
It does. Picture a thread being sewn through cloth. In, out, in, out. 7 passes through the same membrane, all by one continuous thread.
Why 7? Is there something special about 7?
Something very concrete, actually. 7 is odd.
Meaning the 2 ends finish on opposite sides.
Exactly that. Go in and out an even number of times and you come back where you started. Odd number and you end up across from where you began. So the front end of the chain hangs outside the cell and the tail end hangs inside it.
That's not a fact to memorize. That's just counting.
The counting gets you halfway. It guarantees the ends are on opposite sides. Which end is something we measured, not something we deduced. And it's the front end that's outside. Now look at what that gives you for free. The outside end is where the ligand lands. Adrenaline, histamine, acetylcholine. Whatever the signal is, it arrives out there.
What about the inside end?
The inside end and the loops near it are the docking surface. That's where the G protein comes to sit. So the receptor's two jobs are on the two ends, and the arithmetic put them there.
Okay, so what does the receptor actually do when the ligand lands?
It changes shape. That's it.
That's it?
That's the whole repertoire. The 7 helices shift against each other and the surface on the inside of the cell opens up into a pocket. Nothing else happens.
No chemistry at all?
It makes and breaks no bonds. That's what people mean when they say AGPCR isn't an enzyme. It has no catalytic activity of its own. It cuts nothing, joins nothing, burns no ATP. It's also not an ion channel, so nothing flows through it.
Wait, then it can't be the thing carrying the message inside.
Say more. Why not?
Because a message inside the cell has to get somewhere. Something has to physically arrive at whatever's going to respond. And if all the receptor does is flex in place.
Keep going. There's a bigger reason, and you already know it.
It's sewn in. 7 passes through the membrane means it's stitched into the wall. It can't go anywhere.
There it is. That is the single most useful sentence in this entire topic, and I want to say it slowly.
Go ahead.
The receptor is threaded through the membrane 7 times, so it stays in the wall. It can flex, and that flex genuinely does carry information from the outer face to the inner face. But that's the end of what it can do alone. It can't leave, it can't be in 2 places, and it can't multiply.
So it knows something and can't do anything with it.
It can hand it over. That's the one move it has. And whatever it hands it to has to be able to let go and go somewhere.
That's the G protein.
That's why there's a G protein at all. The 3 subunits, the nucleotide, the timer. All of it comes out of solving that one problem. The receptor knows something and by itself can only stand there holding the news.
One clarification, because I want this right. When you say go somewhere, how far?
Good. Don't let me overstate it. Not off into the middle of the cell. It stays stuck to the inner face of the membrane and slides along it. But sliding along that surface is enough to reach effectors the receptor isn't touching, and to reach more than one of them.
So the handoff isn't about distance. It's about being able to let go at all.
Being able to let go at all. That's the whole difference between a fixed part and a messenger.
That's a much better way in than alpha. One activates GQ.
It's the same fact. One version you memorize, the other you derive. Only one of them survives an exam room at hour 6. Part 2. The G protein. And this one is not a single chain. It's 3 different proteins clipped together.
3 subunits.
Called alpha, beta, and gamma. That's why the full name is a heterotrimeric G protein. Hetero means they're different from each other. Trimer means there are 3.
So alpha, beta, gamma are pieces of one complex, not 3 separate systems.
Pieces of one complex. And here's the practical shortcut. Beta and gamma hold onto each other so tightly that they behave as one object. Nobody talks about beta alone or gamma alone. You'll always hear beta, gamma said as one word.
So functionally it's 2 parts, alpha and beta, gamma.
2 parts. And that simplification costs you nothing, so take it.
Why is it called a G protein? Where's the G from?
From guanine. It's short for guanine nucleotide binding protein. It's named for what it's holding.
Holding what? GTP.
GTP or GDP. It's always holding one of the 2, and which one it's holding is the whole mechanism. We'll get there. Right now, I want its address.
Where does it live?
On the inner face of the membrane, the cytoplasmic side. And this is the part people skip, so listen for the difference. It does not go through the membrane.
It's not a transmembrane protein.
Not at all. It's stuck onto the inner surface, like a magnet on the inside of a fridge door. The way it sticks is chemical. The alpha subunit carries fatty acid tails, and the gamma subunit carries a lipid tail of its own. And those tails dip into the membrane and hold the complex against it.
Greasy little anchors.
Greasy little anchors. And now compare the 2 parts we've met.
The receptor is threaded through. The G protein is stuck on.
So what can the G protein do that the receptor can't?
Let go. It can slide along the inside of the membrane, and the pieces can come apart from each other.
Both. It slides, and the subunits release each other while all of them stay tethered to that inner face. That is not a coincidence or a detail. That is why the message gets handed to this molecule and not some other one. The receptor can't move, so the job went to something that can.
Structure, then function.
Every single time. Tell me where a molecule is anchored, and I can usually tell you what it's allowed to do. Part 3. The effector. This is the enzyme that actually builds or destroys something.
So, this is the one that makes the product.
This is the only one that makes a product. And in the exam-relevant world, there are 2 of them worth knowing by name.
Which 2?
Adenylate cyclase, which takes ATP and turns it into cyclic AMP, and Phospholipase C, which cuts a specific fat in the membrane in half.
Where do those sit?
Both at the membrane, on or against the inner face. Which makes sense, because the alpha subunit that comes to switch them on is crawling along that same surface. Everything in this system happens on one plane.
So nothing has to travel far.
Nothing except the products, which turns out to matter enormously, remember that.
Let me say the 3 back before we go on.
Please.
1. The receptor. A single chain, 7 passes through the membrane, locked in place, no chemistry. Its only move is changing shape.
2.
2. The G protein. 3 subunits, alpha plus a beta-gamma pair, sitting on the inner face of the membrane on lipid anchors, and able to come apart and move.
3.
3. The effector. An enzyme at the membrane. Adenylate cyclase or Phospholipase C. The one that manufactures.
That's the whole cast. And notice what we still haven't said. We haven't said what any of it does. We've only said what each thing is and where it lives.
And now the doing part gets easier.
Now the doing part is almost forced. Open up the G protein. 3 subunits. Which one is the worker?
You said alpha holds the GTP.
Alpha holds the nucleotide, and alpha is the one that leaves and goes to touch the enzyme. Alpha is the executive.
Is beta gamma just structural, then?
At the level this question is asking, treat beta gamma as the piece that holds alpha in the off position. When alpha is clipped to beta gamma, alpha is not working.
So beta gamma is a clamp.
A clamp is a fair first model. It's not the whole truth, and I'll come back at the end and show you a real job that beta gamma does on its own, because it explains a classic exam finding. But for now, alpha works, beta gamma holds.
Alpha works. Beta gamma holds.
And that means when you see G's, G, G, Q, G, Q, you should immediately be asking a question.
What's different between them?
What's different between them? Which is the question you actually came here with? Here's the answer, and it's cleaner than most people expect. The little letter, the S, the I, the Q, names the alpha subunit.
So GS and GQ aren't two unrelated proteins.
They're the same chassis with a different alpha in it. GS is alpha S clipped to beta gamma. G is alpha I clipped to beta gamma. GQ is alpha Q clipped to beta gamma.
And the beta gamma part?
We're going to hold beta gamma constant. There are several beta and gamma versions, and they aren't pure filler. But none of that changes the answer to anything you'll be asked. Treat beta gamma as the shared part.
One design, 3 interchangeable alphas.
One design, 3 engines. And now the payoff. The alpha is the piece that detaches and goes to find an enzyme, right?
Right.
So whichever alpha you've got determines which enzyme gets visited. Alpha S goes to adenylate cyclase and switches it on. Alpha I goes to the same enzyme and switches it off. Alpha Q goes somewhere different, to Phospholipase C, and switches that on.
So the alpha type is a whole downstream story.
The alpha type is the entire downstream story. The receptor decides nothing about what happens next. The receptor only decides which G protein gets grabbed. After that, alpha runs the show.
So, what is GQ has a real answer now.
Say it.
GQ is a 3 subunit complex sitting on the inner face of the membrane, containing the Q-type alpha subunit, and it is a completely separate protein from the receptor.
That's it. Not a piece of the receptor, not a domain of the receptor. A different molecule that the receptor grabs and lets go of. Now 2 traps, and they're both traps of language rather than biology. First one.
Go.
People say things like the I receptor or the S receptors, as if he named a class of receptor. It doesn't. The S, the I, and the Q name an alpha subunit and nothing else.
What's the correct sentence?
The M2 receptor couples to G. The receptor has its own name, M2, and separately it happens to plug into the I-type G protein.
Receptor names and G protein names are 2 different naming systems. Then,
2 different systems that meet in a table. And while we're here, the memberships, because this is where people get it backwards.
which ones are G?
Only 3. Alpha, 2, M2, and D2.
What about beta 2, H2, V2?
All three of those are Gs. That's the classic mix-up, and it's worth saying out loud. The number 2 on the end tells you nothing on its own. Every G receptor happens to carry a 2, but plenty of 2s are Gs.
A hint, then not a rule.
A hint you can use in one direction only. We'll set up the full memory system shortly. Second trap, and this one quietly wrecks people's understanding for years.
Worse than the first?
Sneakier. Say this phrase out loud. The alpha, one receptor couples to GQ, which contains the alpha Q subunit.
That sentence has 2 alphas in it.
2 alphas that have nothing to do with each other.
Okay, unpack that.
The alpha in alpha-1 receptor is a classification of adrenergic receptors. Alpha type versus beta type. It describes what the receptor looks like and which drugs bind it. It lives entirely on the outside surface of this system.
What about the other alpha?
The alpha subunit is a physical piece of a completely different protein sitting on the inside of the membrane. Different molecule, different gene, different side of the wall.
They just collided on a Greek ladder.
Pure accident of naming. Rule of thumb. Alpha 1, alpha 2, beta 1, beta 2 with numbers after them are always receptors. Alpha S, alpha I, alpha Q and beta gamma are always pieces of the G protein.
Numbers mean receptor. Letters mean subunit.
Close enough to be useful, and it'll save you.
Here's something I've never been clear on. What is the G protein actually for? Is it holding the receptor in place? Is it some kind of scaffold?
No, and I love that question, because the answer is so much better than scaffold.
It's not structural at all. It's not structural.
It holds nothing up. It anchors nothing. It has no architectural job whatsoever. The G protein is a switch with a built-in timer.
A timer.
Think about what the cell actually needs here. A hormone shows up for a moment. That's an event. It happens and it's over. But the cell needs a response, and a response has to last long enough to be useful. Seconds, not microseconds.
Something has to convert a moment into a duration.
That's the G protein's entire job description. It takes a ligand touched the receptor just now and turns it into this enzyme is switched on for the next several seconds. And then it shuts itself off without being told to.
It turns itself off.
All by itself. Watch. 6 steps. Step 1 is the resting state.
Nothing happening.
Nothing happening. Alpha is holding GDP, guanosine diphosphate, 2 phosphates. And when alpha is holding GDP, its shape is one that beta gamma likes, so the 3 subunits are clipped together.
So the complex is intact.
Intact and idle. It's drifting around on the inner face of the membrane, bumping into things, doing nothing.
And GDP means off.
GDP means off. 2 phosphates, switch is down. Remember it that way. It's the lower number and the lower state. Step 2. The ligand arrives on the outside.
In our question, that's phenylephrine.
In our question, that's phenylephrine landing on an alpha, 1 receptor, and the moment it binds, those 7 helices shift against each other.
The shape change.
The shape change. And it propagates. The movement out on the ligand side gets transmitted through the helices to the other end of the protein, and down on the inside face, a pocket opens up.
A pocket. For what? A pocket.
For the G protein. The receptor is now displaying a surface it wasn't displaying a second ago. A passing G protein bumps into it and gets caught. That's the coupling. That's the entire physical meaning of the word. Step 3. And this is the step people never quite get told properly.
What does the receptor actually do to the G protein?
It pries a lid open. The alpha subunit has GDP buried in a pocket. The receptor grabs alpha and levers that pocket open, and GDP falls out.
That's it? It just opens the lid?
That's it. No chemistry, no energy spent. It's mechanical. And this is precisely why the receptor isn't an enzyme. It never changes a bond in anything. It opens a pocket.
Now the pocket is empty. What fills it?
GTP, 3 phosphates.
Why GTP and not another GDP? What makes it pick?
It doesn't pick. That's the beautiful part.
Then what decides?
The ratio in the room. The pocket is picky about one thing. It wants a guanine nucleotide, so ATP is never getting in. But between GDP and GTP, it doesn't discriminate, and the cell keeps far more GTP around than GDP. Textbooks put it near 10 to 1.
The cell doesn't choose. Then it stacked the deck.
It stacked the deck and let the odds do the work. No sensor, no proofreading, no energy spent. Open the lid in a room that's mostly GTP, and GTP is what you get. Half of cell biology is built like this, honestly. Step 4. GTP is now in the pocket. 3 phosphates instead of 2.
1 extra phosphate. Does that really change anything?
It changes the shape of alpha. That extra phosphate group is bulky and charged, and when it settles into the pocket, parts of alpha have to move to accommodate it.
And beta-gamma stops fitting.
Beta-gamma no longer recognizes the surface it was clipped to, and it lets go. The complex comes apart. Alpha with GTP goes one way, beta-gamma goes the other.
That's the switch flipping.
That is the exact instant the switch flips to on. Not when the ligand bound, not when the receptor changed shape. The switch is on when alpha, carrying GTP, becomes a free molecule. Everything before that was set up. Step 5. Free alpha holding GTP slides along the inner face of the membrane until it finds its enzyme.
Which enzyme depends on which alpha?
Which is the whole point of the last 20 minutes. Alpha C finds adenylate cyclase and turns it on. Alpha C finds the same enzyme and turns it off. Alpha CQ finds phospholipase C and turns it on.
Hang on. Alpha S and alpha I go to the same enzyme?
The very same enzyme. One tells it to run, the other tells it to stop.
That's a strange design.
It's an efficient one. You don't need a separate machine to lower cyclic AMP. You need a way to lean on the machine you already have, and it gives you a 2-for-1 in the naming, which I'll take.
Meaning?
S is for stimulatory. I is for inhibitory. The letters are the answer. You don't memorize what alpha S does, you read it.
And Q?
Q stands for nothing helpful, which means Q is the only one of the three you actually have to sit down and learn. One item, that's the price of admission for the entire roadmap. Step 6, and this is my favourite piece of the design.
How does it stop?
Nobody stops it. It stops itself. The alpha subunit is not only a GTP holder. Alpha is itself an enzyme. Specifically, it's a GTPase.
So it chops the phosphate of its own GTP.
Off its own. It's holding the thing that keeps it switched on, and it slowly destroys it. GTP becomes GDP back down to 2 phosphates.
Then the shape reverts.
Shape reverts, beta-gamma recognizes it again and clips back on, and we're standing in step 1. The switch is off, nothing external was required.
So the length of the signal is just how long alpha takes to cut its own GTP.
That's the timer. Seconds, give or take. And there's a family of proteins called RGS proteins, whose entire job is to hurry that hydrolysis along, which is how a cell shortens a signal when it wants to.
A self-destructing on switch.
A self-destructing on switch, which, when you think about the alternative, is exactly what you want. Imagine a signalling system that needed a second signal to turn the first one off.
You'd need an off switch for the off switch.
Forever. So instead, every switch here is born with a fuse already lit.
Can I ask the annoying question? Why all this? Why not have the receptor just touch the enzyme directly and skip the middleman?
2 reasons, and the first one is amplification.
Because of the handoff?
Because the receptor stays active for a while, and during that while, it doesn't grab one G protein. It grabs one, opens its lid, lets it go, grabs another, opens that one, lets it go. It keeps doing that as long as the ligand is sitting on it.
So one ligand molecule switches on a whole pile of G proteins.
A whole pile. And then each of those alphas switches on an enzyme. And each of those enzymes keeps producing second messenger the entire time it's on.
It multiplies at every layer.
Multiplies at every layer. That's how a hormone present at a vanishingly low concentration in your blood produces a response you can measure across a whole tissue. Direct contact would buy you 1 for 1. This buys you 1 for a great many, and it does it at every layer.
What's the second reason?
The second reason is the more interesting one, and it deserves its own chapter. Count the signals your cells have to recognize. Adrenaline, histamine, acetylcholine, vasopressin, dopamine, dozens of peptide hormones, light in your retina, smells in your nose.
Hundreds of different molecules.
Hundreds. So you need hundreds of different receptors because recognition has to be specific. A receptor that can't tell histamine from adrenaline is useless.
Fine. So we need lots of doors.
Lots of doors. Now count what happens inside. How many different things can the inside of a cell actually do in response?
Fewer. I assume.
3. Raise cyclic AMP. Lower cyclic AMP. Raise calcium.
That's it? 3?
For the pathways you'll be tested on, 3. There's a 4th family, G1213, doing other work, and I'm setting it aside on purpose. So the architecture has to solve a shape problem. Hundreds of doors on the outside, 3 corridors on the inside.
And the alpha subunit is the connector.
The alpha subunit is the adapter plug. Hundreds of receptors, 3 alphas, 3 corridors. Every receptor's job is to pick an adapter. Corridor 1 and corridor 2 share an enzyme, which we said already, Adenylate cyclase, converting ATP into cyclic AMP.
And alpha S turns it up. Alpha I turns it down.
Stimulatory and inhibitory. And cyclic AMP then goes and activates protein kinase A, which is where that whole branch ends up.
So if I know a receptor is GS, I know the endpoint without thinking.
Cyclic AMP up, protein kinase A on. And if it's G, cyclic AMP down. 2 corridors, 0 memory work, because the letters told you.
Which leaves Q.
Which leaves Q, corridor 3. Alpha Q switches on phospholipase C, and phospholipase C is going to hand us calcium.
And Q's letter tells me nothing.
Nothing at all. So here is the honest accounting for this whole topic. Out of 3 corridors, you have to learn 1. Out of everything downstream of alpha, you have to learn nothing because it's fixed.
So what's actually left to memorize?
1 column. Which receptor plugs into which alpha?
And that part has no logic to it?
None that helps you. It's an evolutionary accident. There's no rule that says histamine receptors ought to be GQ. H1 is GQ and H2 is Gs, and no amount of reasoning gets you there.
I appreciate you saying that out loud. Honestly.
It matters though. If you know which part is arbitrary, you stop wasting effort trying to understand it, and you stop distrusting your understanding when it fails to explain it.
Memorize the arbitrary column. Derive everything else.
Everything else. And the column is small. Learn the GQ list, learn the GI list, and everything unclaimed is Gs.
Give me the GQ list.
5 members. H1, alpha, 1V1, M1 and M3. There's a phrase people use for it. Have 1 M and M, HAV1MM.
H1 alpha 1 V1 M1 M3. And G?
3 members. Alpha 2, M2, D2.
And then anything left on this list is Gs.
Beta 1, beta 2, beta 3, D1, H2, V2, all Gs. And be careful with the word list. This is the standard autonomic set the exam actually asks about, not a census of every receptor in the body. There are plenty more, and some couple to more than 1 alpha, depending on the tissue.
But for exam purposes, 8 items.
8 items memorized, total. That's the entire non-derivable content of this topic.
Let me say all 3 corridors back. And I'll say them without looking.
Go.
GS. FS turns on adenylate cyclase. Cyclic. AMP goes up. Protein kinase A switches on.
2.
GILAI turns off the same enzyme. Cyclic AMP goes down.
3.
GQ. Alpha Q turns on phospholipase C. And the phospholipase C leads to calcium.
And which one is our question on?
Phenylephrine hits alpha 1. Alpha 1 is on the GQ list, so we're on corridor 3.
Corridor 3 the whole way, which means for the rest of this episode, cyclic AMP is not our business. Keep that. It's going to hand you 2 wrong answers for free. So alpha Q has switched on phospholipase C. Phospholipase C is going to cut something. Before we watch it cut, we have to look at what it's cutting, because everything that follows comes out of that molecule's shape.
Structure first.
Structure first. The target is called IP2. And I want you to picture it as a lollipop stuck sideways into a wall.
Okay. Describe the lollipop.
Two ends, and they could not be more different. On one end, there's a ring-shaped sugar called inositol, and hanging off that ring are phosphate groups. Phosphates carry negative charge, so that end is charged, and charged means it loves water.
That end can't be inside a membrane.
It refuses to be, so it sticks out of the membrane's inner surface into the watery cytoplasm. That's the candy head, poking into the room.
And the other end?
The other end is two long fatty acid chains. Pure grease. They won't go near water, so they stay buried in the middle of the membrane. That's the stick driven into the wall.
One molecule. Half of it in the wall, half of it sticking out.
And the two halves are held together by a small linker in between, which is exactly where the knife goes. Phospholipase C arrives and cuts right at the neck. One cut. The molecule becomes two molecules.
One enzyme, one cut, two products.
Two products. And this is the part that makes the whole pathway make sense. Give them their names first. The candy head becomes IP3, inositol trisphosphate. The greasy stick becomes DAG diacyl glycerol.
Two names I've definitely memorized and definitely lost.
You won't lose them again, because you're about to derive where each one goes. Don't tell me what they do. Tell me what they are, and then tell me what they're allowed to do.
IP3 is the charged half.
So
IP3 is charged, which means it's water-soluble, which means it can't stay in a membrane. It has to leave.
It has no choice. The instant it's cut free, it's floating in the cytoplasm.
And DAG is a greasy half, so DAG can't leave. It's stuck in the membrane where it was.
Neither of them is making a decision. Chemistry made both decisions before the cut happened. Now push it one step further, because this is where you get two facts for free.
Go on.
IP3 is now travelling. If it's travelling, where does its target have to be?
Somewhere else. Not at the membrane it came from.
Somewhere else in the cell. Solubility tells you it's free to be somewhere else, and where it actually turns out to be is a measured fact on the endoplasmic reticulum deep inside. So its receptor gets called an intracellular receptor. That word in an exam question isn't decoration. It's telling you which of the two products you're following.
What's the free fact for DAG?
DAG can't travel. So anything that wants to use DAG has to come to it.
The target has to walk over to the membrane.
The target has to relocate, and it does. We'll watch it happen in a minute. Two products, two solubilities, and the entire downstream geography falls out of them. Nothing here was memorized. Follow IP3. It drifts across the cytoplasm and arrives at the endoplasmic reticulum. In a muscle cell, we call that same compartment the sarcoplasmic reticulum.
Which is the calcium warehouse.
The calcium warehouse. The cell keeps calcium locked in there at high concentration and keeps the cytoplasm almost empty of it, on purpose.
Why on purpose?
Because a signal has to be a change. If calcium were everywhere all the time, releasing more of it would say nothing. Keep the room empty, and even a small release is deafening.
IP3 arrives and knocks.
IP3 binds to a protein in that membrane called the IP3 receptor. And here's the structural detail worth having. That receptor isn't a passive binding site. The IP3 receptor is itself a channel, a gate for calcium held shut.
So binding opens it.
IP3 binding drives the gate open and calcium pours out of the store into the cytoplasm. A messenger arrives, a gate opens, and the room fills with calcium.
This receptor is the thing our drug blocks.
This is the thing our drug blocks. Remember exactly where we're standing.
Calcium's out. What does it do?
In a smooth muscle cell, calcium doesn't act on the contractile machinery directly. It has to be handed to a sensor first.
In skeletal muscle, that sensor is troponin.
In skeletal and cardiac muscle, yes. Smooth muscle has no troponin at all.
So what does smooth muscle use?
A protein called calmodulin. And that name is doing you a favour. Cal, for calcium modulin, for modulating. It's a calcium modulating protein, and it says so.
Calcium binds calmodulin. And then?
The pair switches on an enzyme called myosin light chain kinase. That kinase adds a phosphate to myosin. Myosin starts pulling on actin, and the cell shortens.
And in a blood vessel, cells shortening means?
The vessel narrows. Blood pressure goes up, which is why phenylephrine is a blood pressure drug and why it's in your decongestant spray. Narrow the vessels in a swollen nose and the swelling comes down.
So that's the IP3 leg finished. What about DAG, still sitting in the membrane?
DAG is waiting for someone. And the someone is protein kinase C.
Where does protein kinase C live?
Floating in the cytoplasm, doing nothing until it's called. And to be called, it needs two things to be true at once.
2.
1. There has to be DAG in the membrane. And since DAG can't move, that means protein kinase C has to physically relocate. It travels to the inner face of the membrane to reach it.
That's the fact you promised earlier.
That's the one. It was forced by DAG being greasy. And 2, it needs calcium.
It needs the product of both legs.
Both legs. DAG from one, calcium from the other. Look at the shape of what we've built. One enzyme cuts one molecule, the two halves go in opposite directions, and then they come back together at a single point.
And that single point is protein kinase C.
That single point is protein kinase C. It's the only thing in this entire pathway that requires two separate inputs to be present at the same time.
Which makes it the fragile spot.
Cut either leg, either one, and the thing that fails is the meeting point, not the leg you cut.
That sounds like it's about to be the answer to something.
Hold that thought for about 4 more minutes. One honest footnote, because I don't want to hand you something too clean.
Go ahead.
Protein kinase C isn't one enzyme. It's a family, and the members have different requirements. The classic ones. The group called conventional protein kinase C need both calcium and DAG, exactly as I described.
And the others?
There's a second group that needs DAG but manages without calcium, and a third that needs neither.
So protein kinase C needs calcium. It's true of the conventional group.
True of the conventional group, which is the group these questions are built on. And strictly it wants membrane phospholipid too. The exam shorthand is calcium plus DAG, and that shorthand is what's being tested. So the answer doesn't move. But now you know the shape of the exception, and an isoform in a question won't throw you.
Let me run the whole arm without stopping.
Please.
Alpha Q switches on phospholipase C. Phospholipase C cuts IP2 at the neck. The charged head floats off as IP3. The greasy tail stays behind as DAG.
Keep going.
IP3 crosses the cytoplasm to the sarcoplasmic reticulum and binds the IP3 receptor, which is the calcium gate. The gate opens, calcium floods out.
And the calcium does two jobs.
Calcium goes to calmodulin, which switches on myosin light chain kinase, and the muscle contracts. And calcium also goes to protein kinase C, which is waiting at the membrane with the DAG.
And protein kinase C needs
Both calcium and DAG together.
That's the arm. Every step in it is something you can rebuild from where a molecule sits and whether it's greasy or charged, which is the whole reason we spent 30 minutes on parts before we drew a single arrow. Here's the question we've been building toward, and I want you to notice how little of it you now have to read carefully.
Let's have it.
A pharmacology lab is testing a new tool compound. The compound has exactly one job. It parks on the IP3 receptor and stops IP3 from binding to it.
The gate on the calcium store.
That gate. And the question is explicit that the compound does nothing else. It doesn't touch any surface receptor. It doesn't touch any G protein. It doesn't touch any enzyme.
So we know precisely where the block is.
Precisely. Now the setup. Cultured aortic smooth muscle cells in 2 dishes. One dish gets pre-treated with the compound. The other gets nothing.
Then what?
Both dishes get the same stimulus. Phenylephrine.
Both dishes. So the stimulus isn't the variable.
The only variable is the block. Then they measure the activity of 5 enzymes in both dishes, and they ask you which one has fallen in the treated dish. 5 enzymes. Listen to all of them before you decide.
Go.
A. Adenylate cyclase. B. Lipoxygenase. C. Phosphodiesterase. D. Phospholipase. C. E. Protein kinase. C.
Say them once more. Slowly. For anyone listening rather than watching.
A. Adenylate cyclase. B. Lipoxygenase. C. Phosphodiesterase. D. Phospholipase C. E. Protein kinase C.
Pause it here. Pick one. Say it out loud, because saying it out loud is what makes you commit, and committing is what makes the answer stick.
The answer is E. Protein kinase C.
The convergence point.
The convergence point, the only enzyme in the pathway that needs 2 things at once, and we just took one of them away. Now let's do the part that actually earns you marks. Let's walk it, and then let's kill the other 4 properly. Start at the top, and don't skip. Phenylephrine. What is it?
A. Selective alpha. 1. Agonist.
Alpha. 1 is on which list?
The GQ list. Have 1 M and M.
So, which corridor are we in?
Corridor 3. Alpha Q switches on phospholipase C, which cuts IP2 into IP3 and DAG.
Both products made?
Both. Nothing has been blocked yet at that point.
Now, put the drug in. Where exactly does the block sit?
Downstream of both products. It's on the IP3 receptor, which is the next step after IP3 has already been made and has already traveled.
So what fails?
The gate doesn't open. Calcium stays locked in the store. So there's no rise in cytoplasmic calcium.
And what survives?
DAG. Completely untouched, still sitting in the membrane, made in normal amounts.
So now name the casualty.
Protein kinase C. It's got its DAG. It never gets its calcium. And it needs both. It's the only thing that cares that one leg went missing.
That's the answer. Derived in about 40 seconds. Nothing memorized. Now, the wrong answer that catches the most people, and it catches good students specifically,
D. Phospholipase C.
D. And I want to be fair to it, because the reasoning behind picking it isn't stupid. It goes like this. The pathway has been disrupted. Phospholipase C is the central enzyme of that pathway. Therefore, phospholipase C must be affected.
Which sounds reasonable until you ask where things are.
Until you ask about direction. So ask it. Where is phospholipase C relative to the block?
Upstream. Well, upstream. It acts before IP3 even exists.
And what does the drug physically touch?
The IP3 receptor. Nothing else. It never goes near phospholipase C.
So does phospholipase C have any way of knowing that something failed downstream?
No. It has no idea. It's still being driven by alpha Q. It's still sitting in the membrane. It's still cutting IP2.
It's working, and it's oblivious. So here's the rule, and then, straight after, the honest limit on the rule, because I'm not going to hand you a law that isn't one.
Rule first.
In the feed-forward pathway this question is testing, a blocker lowers what sits downstream of it, not what sits upstream. The diagram runs one way. A jam at the exit doesn't slow the factory.
What's the limit?
Real cells have feedback. Calcium genuinely does feedback onto phospholipase C. So in a living cell, taking calcium away can drag phospholipase C down a little as well. There are other loops too.
Doesn't that wreck the answer?
It doesn't. And the reason is in how the question is worded. It asks which enzyme is most likely to fall. Protein kinase C loses one of the two inputs it requires, direct and large. Phospholipase C loses a feedback nudge, while alpha Q is still driving it the whole time, indirect and small.
So the exam rule holds. I just shouldn't turn it into a law of nature.
Use it to sort 5 options in 20 seconds. Don't go telling a biochemist that downstream can never touch upstream. Both things are true, and knowing which one you're standing on is the actual skill.
So when I see which enzyme decreases, I find the block first and then look only to the right of it.
Find the block, mark it, cross out everything to its left before you even read the options. Half of these questions collapse the instant you do that. Now A and C, and these two die together for the same reason.
A was adenylate cyclase. C was phosphodiesterase.
What corridor do those two belong to?
the cyclic AMP corridor. Adenylate cyclase makes cyclic AMP. Phosphodiesterase destroys it.
And which G protein runs that corridor?
GS and GI, not GQ.
And our stimulus is phenylephrine on alpha 1, which is
GQ. So we never even entered that corridor. Those two enzymes weren't switched on to begin with, so there's nothing to decrease.
That's why I told you 30 minutes ago to remember we were on corridor 3. 2 free eliminations, and you didn't have to think about either of them.
That's a nice return on one sentence.
That's the return you get on knowing which road you're on. And it's worth adding one thing about phosphodiesterase, because it shows up constantly in pharmacology. It's the enzyme that ends these signals. Milrinone blocks the type that breaks down cyclic AMP. Sildenafil blocks a different type, and that one guards cyclic GMP, not cyclic AMP. Both work the same way. Stop the disposal and the messenger lasts longer. B. Lipoxygenase. Different kind of wrong.
It's the leukotriene enzyme.
It is, and the line it belongs to starts somewhere else entirely. It starts with phospholipase A2, which cuts a different bond in a membrane phospholipid and releases arachidonic acid.
Then arachidonic acid splits 2 ways.
Cyclooxygenase takes it toward prostaglandins. Lipoxygenase takes it toward leukotrienes. That's the inflammation branch. Steroids block phospholipase A2 at the top, which is why they shut down both.
Different enzyme, different substrate bond, different products.
And here's the memory hook I actually want you to keep, because it's not something I invented, it's just the letters. Phospholipase A gives you arachidonic acid. Phospholipase C gives you calcium.
A for arachidonic, C for calcium.
And while we're collecting Cs, protein kinase C needs calcium, and calmodulin is named for calcium. Every C in this pathway is pointing at the same element.
One honest check, though, doesn't DAG also feed into arachidonic acid?
It can, through a different enzyme. Good catch. But look at what our drug did to DAG.
Nothing. DAG production is unchanged.
So even by that route, there's no reason for lipoxygenase to fall. The claim isn't that DAG and arachidonic acid never touch. The claim is that nothing in this experiment reduced either one. Last thing about the question itself, and it's the piece of craft I admire in it.
Which is?
Read the question again in your head, from the first word to the last option. The word calcium is never in it, not once.
And calcium is the entire mechanism.
Calcium is the entire mechanism, and you have to supply it yourself. The question hands you IP3 receptor and expects you to know that the IP3 receptor is a calcium gate.
So if you learned IP3 causes calcium release as a slogan, you can still miss it.
You can still miss it because a slogan doesn't tell you the receptor is a channel. Knowing the structure does. That's not an accident of this one question. That's what the whole exam is doing. Now let me show you what you actually bought with the last 50 minutes, because it's much bigger than one question.
Go.
Adrenergic receptors. There are 4 you care about. Alpha 1, alpha 2, beta 1, beta 2. And here's the strange part. Your body stimulates all four with essentially the same molecule.
Noradrenaline and adrenaline.
Same messenger, 4 receptors. So how do you get 4 different outcomes from one molecule?
Different adapters. Each receptor plugs into a different alpha.
Watch it happen. Alpha 1 is GQ, so it raises calcium, and in a vessel wall that means the vessel squeezes. Beta 1 is Gs, so cyclic AMP goes up. And in the heart, that means faster and harder beats.
And beta 2?
Also Gs, also cyclic AMP up, but in bronchial smooth muscle, the effect is relaxation, which is why your asthma inhaler is a beta 2 drug.
Wait, beta 2 raises cyclic AMP and relaxes muscle, but alpha 1 raises calcium and contracts it.
Same organ system, opposite outcomes. 1 hormone, and alpha 2 is G, which lowers cyclic AMP, and it mostly sits on the nerve terminal telling it to stop releasing more.
The receptor subtype is doing all of the work.
All of it. The ligand is just the ligand. So now tell me what a selective drug actually is.
It's a molecule that picks one receptor subtype.
And by picking a subtype, what has it really picked?
A corridor. It's chosen which intracellular road gets used.
That's it. That is the whole business. Phenylephrine picks alpha, 1 and nothing else, which means it presses the GQ switch and only the GQ switch, which means calcium, which means vasoconstriction, which means blood pressure up and a nose that stops running.
And it never touches cyclic AMP.
Never goes near it. Which is exactly why 2 of our 5 options were dead on arrival.
One detail I like. The Chinese name for phenylephrine literally means deoxyadrenaline.
And the name is telling you something real. It's adrenaline missing 1 hydroxyl group off the ring. Losing that group means the enzyme that normally chews up adrenaline can't grab it as well. So given intravenously, it lasts longer than adrenaline does.
Given intravenously. You said that deliberately.
I did. Because swallowed is a different story and it's worth knowing. Oral phenylephrine gets taken apart in the gut wall and the liver before it reaches the circulation, and the FDA has proposed pulling it from the over-the-counter monograph as an oral decongestant on the grounds that it doesn't work.
So the tablet and the nasal spray aren't the same drug story.
Same molecule, completely different fate. And the mechanism we just built is exactly why you can hold both facts at once. Receptor pharmacology tells you what it does when it arrives, and it says nothing about whether it arrives. I owe you something from earlier. I said beta-gamma was a clamp and told you it wasn't the whole truth.
You did.
When the complex splits, beta-gamma is also released. And it isn't inert. It can go and do things of its own.
Like what?
Here's the one worth knowing. Your vagus nerve releases acetylcholine onto the pacemaker cells of your heart. It lands on an M2 receptor. M2 is which G protein?
GI. It's on the list of 3.
So cyclic AMP goes down, and that slows the heart. True, and most people stop there. But the beta-gamma that got released does something else. It goes and opens potassium channels.
Potassium leaving the cell.
Potassium leaves, the cell gets more negative. and a more negative pacemaker cell is a slower pacemaker cell.
Vagal slowing is two mechanisms, not one.
Two mechanisms, and the second one is beta-gamma acting directly. Keep it, because it's the standard exam-level example, and because it stops you thinking of beta-gamma as packing material. Last piece, and this is how you find out whether the switch cycle really went in.
A test.
Two diseases, cholera and whooping cough. Both toxins attack this system at opposite ends and both end up raising cyclic AMP. Explain that and you understand the cycle.
Start with cholera.
Cholera toxin chemically modifies alpha S and destroys its ability to hydrolyze GTP.
So it breaks step 6, the self-shutoff.
Breaks the fuse. Alpha switches on normally and then cannot switch itself off, ever. So adenylate cyclase runs continuously. Cyclic AMP climbs and stays climbed.
And in the gut, that means?
Chloride and water pumped into the lumen without stopping. Liters of watery diarrhea. It's one broken timer, scaled up to a disease that can kill you in a day.
And pertussis?
Pertussis toxin modifies alpha I, but at the other end of the cycle, it makes alpha I unable to be activated by its receptor. It's stuck holding GDP.
It can never turn on.
Never turns on. And alpha I's job was to press down on adenylate cyclase.
So if the brake never engages, cyclic AMP drifts up.
Cyclic AMP goes up. Two toxins. One jams the off step, the other jams the on step, and both end in the same place for opposite reasons.
If you can say why both raise cyclic AMP, you've understood the cycle.
That's my exam for you. Not mine, really. It's the cycle's exam. Let's change one thing at a time and see what moves. Same experiment, same cells, same phenylephrine.
Change one thing.
Version 1. The drug doesn't block the IP3 receptor. It blocks DAG from binding protein kinase C instead.
Then, protein kinase C still fails because it still loses one of its two inputs. But calcium is fine, so the muscle still contracts.
Good. The same answer, but a different casualty list. Version 2. The drug blocks the alpha, one receptor itself.
Then, nothing downstream happens at all. Phospholipase C activity drops too. This time, because now the block is upstream of it.
So D becomes a right answer.
D becomes a right answer in a question that reads almost identically.
Which is the whole reason we spend time on direction rather than on lists.
Give me one more.
Version 3. Same drug on the IP3 receptor, but the stimulus is now isoproterenol, a beta agonist.
Beta is GS, so we are on the cyclic AMP corridor. And the IP3 receptor was never involved. Blocking it changes nothing.
Nothing at all. Same drug, same cell, zero effect because you changed which door was knocked on. 3 variations, 3 different answers, 1 architecture. Here's how you make this permanent, and it takes about 90 seconds a day for 3 days.
Not rereading it.
Rereading does almost nothing. Rebuilding does everything. So cover all of this up, give yourself one word, phenylephrine, and say 5 sentences out loud.
Let me try them.
Go.
1. Phenylephrine hits offer 1. Offer 1 is on the GQ list, so we're on the GQ corridor.
2.
2. Alpha Q leaves the complex and switches on phospholipase C, which cuts the lollipop in the membrane in half.
3.
3. Charged head flies off as IP3. Greasy stick stays behind as DAG.
4.
4. IP3 crosses to the calcium store and opens the gate because the IP3 receptor is a channel.
5.
5. Calcium goes to calmodulin for contraction, and calcium plus DAG meet at protein kinase C.
5 sentences, whole pathway. And if you stall on one of them, go back and fix only that one. Don't restart from the top. That's the mistake that turns a 90-second drill into a 40-minute reread. One rule to leave with, and it isn't about this pathway.
Say it.
Find the structure first, and the function stops being something you have to remember.
That's what happened here, isn't it? Every single step.
Every step. The receptor crosses the membrane 7 times, so it can't move, so it can't be the messenger. That's why the G protein exists. The head of PIP2 is charged, so it has to leave, so its receptor has to be somewhere else. The tail is greasy, so it has to stay, so protein kinase C has to come to it.
And the answer to the question came out of the shape of the pathway, not out of a fact about the answer.
Protein kinase C is where the 2 legs meet, so protein kinase C is what breaks when you cut one. You needed exactly one fact about it, that it wants calcium and DAG together. Everything after that was geography.
That's the episode. If the pathway held together this time, that's because we built the floor before the building.
And that's how the rest of this series works too. Structure, then function, then the question.
Subscribe if you want the next one. And if a specific step in here still feels shaky, tell me which one in the comments, and I'll build an episode on it.
Go and do the 5-sentence drill. Right now, out loud, before you open anything else. It'll take you 90 seconds and it'll save you an hour later.
See you next time.